So we talked about mechanisms of reproductive isolation last week, and I just want to clear up a few issues that we noticed in your in-class activity, and they mostly have to do with post-zygotic reproductive mechanisms. So hybrid viability is that the hybrid offspring do not actually develop. So even though the male and female can mate and the gametes can fuse together, there's something wrong with the offspring and there's typically a miscarriage, or the offspring dies very, very young. It never lives to be able to reproduce. Usually this happens because there's different numbers of chromosomes between the mother and the father, and the body recognizes it in the female as a foreign invader and aborts it. The other one is hybrid sterility, and that's when they're sterile as adults, which means they do not produce gametes. So when you're sterile, there's no gamete production. So if you're male, you don't produce sperm. If you're female, you do not produce eggs. So that's what it means when you're sterile. Any questions on that? No, okay. So today we're going to talk about evolutionary patterns. So here is different groups of insects. Here we have beetles. They're the most diverse group of insects. And how do we know a beetle isn't a butterfly? Beetles do have wings, but they're covered in a protective coat. So think about a ladybug. Ladybugs can fly. Ladybugs are beetles, but their wings look different, right? We would never think that this type of organism is the same as this type of organism down here. But we do know that beetles and butterflies are more closely related to each other than a beetle is to a human. And so that's what we're going to talk about today. We're going to talk about relationships between different organisms and how they have changed over time. So a phylogeny shows the evolutionary relationships among organisms. So we could think of a phylogeny almost of a family tree of organisms, or like a pedigree. We talked about pedigrees last semester. You could think of it almost like that. The patterns of lineage branching produced by the evolutionary history of the organisms being considered are what goes into a phylogeny. So we're thinking about what makes humans, for example, different than insects. We only have 4 limbs. Insects have 6. We do not have wings. Almost all insects have wings. We have a skeleton internally, inside our body. Insects have a skeleton on the outside of their body. So we can use these different characteristics to figure out evolutionary relationships. So we like to put them on what is called a tree. So how do we read a tree? So we call this a phylogenetic tree or cladistics. And we have our ancestral lineage. So there's some sort of organism back here. This is our root. So if you think the roots of the tree, they ground the tree into the ground. The root is whatever the common ancestor was. So there was some organism that used to live a very long time ago that was the common ancestor of both insects and humans. And then characteristics change. And so then we have branching of our tree. And every time we have a branching area occurred, it's called a node. At the end of our tree are our taxon, or our different organisms that we're interested in looking at. And taxon could be very specific. We could be talking about specific species, or we could be talking about groups of organisms. So we could say we're looking at taxon A through F for beetle species, or we could say taxon A through F are different types of animals. So we can have insects and mammals and reptiles and birds. Taxon that are on, if you pass a node, taxon that are at the tip of one of these nodes are called sister taxa, because they're right next to each other. They're sisters. They're the most closely related to each other compared to all the other organisms on our tree. And then when we don't know the exact evolutionary relationship, so see we have 3 organisms here, we're not sure who is more closely related to each other, we have Platomy. So we don't have enough information to know who is more closely related to who. Yes, ma'am.
Sorry, you said the node were 2 branches of
Yep, so this is a node, that's a node, that's a node.
Okay. So there's a species addition.
So there's some sort of common ancestor, yes.
Okay. But it's unknown.
Most of the time the common ancestor is unknown, yes. So there's another way to look at it. So here I put different organisms. So this isn't specific to species, these are just different types of animals. And we can see here we have a node here. And then over time, characters developed that separated or made these different groups of organisms distinct. So having an exoskeleton, a skeleton on your outside, led to lobsters and spiders. All other animals have vertebrae, so they have a backbone. Lobsters and spiders don't have a backbone. So that separated all these 2 organisms, these sister taxa, from all the other organisms because there was a change in a character state. Here, the main character state that developed were lungs. So we have lungs. Then amniotic eggs develop. So we have eggs that are an amniotic sac. And that's what distinguishes mammals from lizards and birds. Lizards and birds can see UV light. These 2 trees are exactly the same. It is just a different way of looking at them. So I got crafty last night and I made a tree. And if anybody wants pipe cleaners, I have a bunch in my office. You are more than welcome to have some. Okay? These trees are identical to each other. Oh, that's like great music. That's great sound effects. So if you don't believe me, you can take this tree and bend it, and you end up getting the exact same tree with my other pipe cleaners. Don't laugh at my pipe cleaners, they're awesome.
I love this.
My evolution teacher in undergrad showed this to me, I never forgot it. So just by moving the way that we set this up, we now have identical trees. So these are the same things. Remember, guys, I used to be a high school teacher. I love stuff like this. One of the things that we can do with these trees is it doesn't matter which way we look at it. We can look at it this way or this way. It's still showing the same relationships. We still have the root, which I'm holding. We still have our nodes. We still have our sister taxa, which are up here. And what you can do is if you look here, we can rotate around our nodes and we don't change the relationship. So if I rotate these 2 sister taxa just where they happen to appear on the tree, it's still the exact same thing. We still know that the orange and the green organisms are sister taxa at the end of our tree. You can even rotate this one if you wanted to. It's still showing the same relationship. It's still showing that the purple organism diverged first, and then we know that the yellow one diverged next. So even though I twisted it around, it is still the exact same tree. So if you guys want to mess around with these, I'm going to leave them up here, and if you want pipe cleaners, just let me know. You can make them 3D. You can move these around however you want. The relationship does not change. This is still the exact same tree as this. It's just harder to read because it's messy. The out group, in my tree example, is the purple one. Or in here, it's A, and on these trees that are rotated around, it's W. That's a group of organisms that belong in the group whose evolutionary relationship are being investigated. So we're using it, think of it as what we're going to compare everything to. Okay, so it's usually somebody who's way different. We know that they're usually, we like to think of them a lot of times as more ancestral. They have had less changes in allele frequencies over time. So they're the most basal or ancestral organisms. They don't belong to the rest of the group. So we use, in our trees, we have different types of classifications. The first is monophyletic, and that includes organisms from one common ancestor. So if we're looking at our monophyletic tree here, this tree is the same, I just have different parts highlighted. Species 1, 2, 3, and 4 all can be traced back to this one common ancestor. So species 1 and 2 are sister taxa to each other. Species 3 and 4 are sister taxa to each other. So 1 and 2 are more closely related to each other than anybody else on this tree. Species 3 and 4 are more closely related to each other than anybody else on this tree. Species 1, 2, 3, and 4 are more closely related to each other because they share this common ancestor compared to how closely they're related to species 5, 6, and 7. So because we can trace it back to a node and know that there's some common ancestor among these 4 species, they're most closely related to each other. And that's a monophyletic group. So it's a group of organisms that all share the same common ancestor. So mono means one common ancestor. Paraphyletic is a group that includes a common ancestor and some, but not all, of its descendants. So here is our paraphyletic group. Here's our common ancestor that's shared among species 1, 2, and 3, but in this group, we left off species 4. So it's para, it's only part of all of the descendants of that common ancestor. Yeah. So if we were just doing 5, 6, and 7, you can trace them back to all having one common ancestor. So this is a monophyletic group. It's a great question. Polyphyletic does not include one common ancestor. So in a polyphyletic group, typically what happens is we grouped them together because we thought they looked alike or something about them, it was similar. Usually this occurs through convergent evolution, which means there was some sort of environmental factor that caused these organisms to develop similar characteristics even though they're not closely related. So putting butterflies, birds, and bats together because they all have wings would be an example of a polyphyletic group because they do not all share the same common ancestor. Insects have a much different common ancestor than birds and bats do. And birds and bats have a different common ancestor. What we are typically going for when we're thinking about relationships of organisms are monophyletic groups. We want to have what is called a clade. A clade is just a monophyletic group. The reason we want that is because when we're talking about a group of organisms, we want to be talking about organisms that all come from the same common ancestor. There's a question over here.
Yeah, I just wanted to ask, do they all have to be next to each other when you're grouping them? Could you do species 1, 2, and then 6?
Yeah, so you could do species 1, 2, and 6, and that would be a polyphyletic group. Okay. Yep. It was just easier to draw a box around all the ones next to each other. Yes,
how can you do that though? Because if species 3 has a common ancestor, but the sister species is 4, how can that not have the same common as species 3?
So species 5 has a different ancestor.
No, my bad. Sorry. For paraphyletic, and then.
So in paraphyletic, you just left somebody out. So what usually happens in paraphyletic is you didn't realize that one of the organisms you're looking at is actually in that group.
Oh, okay. So it's like theoretical type.
Yes, that would be saying you're looking at mammals and and these are bats, and you didn't realize bats were mammals because they have wings.
Oh.
You left them out. Any other questions? So this is just another way to look at the same information. So we have B and C, these are sister taxa. E and F are sister taxa. G and H are sister taxa. In this example, there is no outgroup because we have organisms that are closely related to each other on both of our branches. Here is our root of our tree, and we have 7 nodes in our tree. So every time there's a branching, we have a node, and there are actually 7 clades. So B and C is a monophyletic group because they share this common ancestor. A, B, and C is a monophyletic group because they share this common ancestor. E and F are monophyletic because they share a common ancestor. G and H are monophyletic because they have a common ancestor. E, F, G, H is monophyletic because they share a common ancestor. D, E, F, G, H, one monophyletic group or clade because they share a common ancestor. And A, B, C, D, E, F, G, H is also one big monophyletic clade because they share a common ancestor. So you can have a clade within a clade, and that's okay. Yeah.
So would if it's A, B, but not C, would that be paraphyletic?
Perfect. So if they said A and B and they ignored C, that would be a paraphyletic group because we're missing one of the descendants. We have some, but not all. Great question.
So in this one, since like they are all traced back to one, could you say that like you wouldn't have a polyphyletic group?
So you could have a polyphyletic group if you left somebody out. So if you said A, B, G, and H, G and H have a common ancestor here, so their clade is basically done, right? And then A and B, their common ancestor is over here. So we're ignoring all these other organisms that come down to the same common ancestor. So that would be an example of polyphyletic because their most recent common ancestor is very different. Yeah.
You took off the A, B, C branches, would D be the out group?
Yes, if you took off A, B, and C and you were just looking at these 5 organisms, D would be considered our out group. Good question. Yeah.
What is D, E, F, G, and H? Like, I know if you left off B, the E, F, G, and H would be your monophyletic group. But how is it not polyphyletic since you left off D and you all share ancestors here? Like,
So-
how are there 2 different ancestors on the same line?
So we have different ancestors here because something happened. There was a change in character state that caused this ancestor to be different than all these other ones.
But it just hit one organism?
Yep, this is just one organism. Say they grew wings. For some reason, there's some mutation, and some of these organisms had wings, and now all of D, whatever species that is, they have wings. None of these organisms have wings. We're actually going to talk about how you make these today as well. So you guys have been asking some really great questions, so here's some real life examples. A paraphyletic group is fish. So if we're looking at this, fish are actually paraphyletic because we're including some but not all of the individuals that have a common ancestor. So fish actually share a common ancestor with amphibians, reptiles, and birds, and mammals. So that's a paraphyletic group. We're missing a bunch of the descendants of that common ancestor because the common ancestor for all fish is way back here. An example of a polyphyletic group are pachyderms, which are elephants, rhinos, tapirs, and hippos. Now, if we look at this phylogenetic tree, here's hippos, here's rhinos, elephants are way up there. They didn't put tapirs, I don't know why, on this picture. But because elephants have a common ancestor here with manatees, rhinos and tapirs have a common ancestor with horses, and hippos actually have a most recent common ancestor with dolphins and whales, they're missing their common ancestor back here, so it's polyphyletic. So what happens usually at polyphyletic groups is they were classified together before we actually understood DNA sequencing. And so we could say, yeah, these organisms all kind of look alike. They're big and cool. So they should all be a group because you would think that a rhino is probably more similar to an elephant than a rhino is to a horse, but that is not true.
Aren't rhinos really fast?
Rhinos are pretty fast, yes. So taxonomy is the science of classifying living things. The classification level is called a taxon. The largest group of organisms that we like to classify are called domains, and there's 3 of them. There's the eukaryotes, there's bacteria, and there's archaea, and we're going to talk about bacteria and archaea next week. So we start with domains. Those are very big classifications. We incorporate a lot of organisms into that. Then we get smaller and smaller. So we start with domains, and then we go, oh, here's domains. Then we have kingdom, phylum. If we're thinking about squirrels, see, we're getting more specific, and we're now in the kingdom Animalia, so all animals. Phylum, chordata, so now we only have organisms that are chordates. Then vertebrata, so you have to have a backbone. Mammalia, so you have to be a mammal. Rodentia, so you have to be a rodent. Then you need to be in a family of squirrels, a genus of these particular type of squirrels. And then finally you get to your species. So it goes domain, kingdom, phylum, subphylum sometimes, class, order, family, genus, species. Okay? So we just go from big ways to classify organisms down to small. So humans, we are in the genus Homo. We are the species sapiens, so we are Homo sapiens. That's what we are. Right. Systematics is what we do with taxonomy. So we take taxonomy, we're classifying these organisms based on characters, and then systematics tries to take those characteristics and mirror the probable evolutionary relationships between these organisms. This is a very old science of trying to classify organisms as being closely related to each other. What used to happen is we would just look at organisms and say, this is what is closely related to other things because they look alike. But now we have molecular analyses that we can use. We can actually analyze DNA and RNA in these organisms and get a better sense of how organisms are closely related to each other, which is how we were able to figure out that birds are more closely related to crocodiles than they are to other reptiles. And a phylogenetic tree, which we were learning how to read right before this, is just a hypothesis. So it's how we hypothesize organisms are related to each other. So if we put taxonomy and systematics together onto a tree, we have a phylum, so a large group. We break into our multiple classes. We then have our orders, families, genera is the plural of genus, and then we get to all of our species that are represented. So that's how we put taxonomy and systematics together. So there's some morphological concepts that we need to know. A homology are 2 or more features that share a common ancestry. Okay, so it's just homology, something about these organisms that show that they share a common ancestry. So for example, if we're looking at this phylogenetic tree, an amniotic egg, so an egg that develops inside a sac, evolved once in all of these organisms, and so that's a homology. It's a homologous trait that we all share. It's how we see that we are more closely related to these organisms than we are to something like an insect. An analogy or an analogous trait is a similar function, but it usually results from convergent evolution. So wings on bats, wings on birds. Here's birds, here's bats. The presence of wings is an analogous trait because it evolved because both these organisms live in the air. They need to fly. But they do not share, just these 2 groups do not share one common ancestor. because all these other organisms are in here as well. So wings had to evolve separately in bats and birds. Yeah, we're gonna skip that one. All right, so you were asking a lot, how do we know what parts of trees are what? We actually can build phylogenetic trees. So our in-group are a group of species or organisms of interest. Our out-group is the species that diverged before the in-group. So it's the most basic, the most basal of all of our organisms we're looking at. We then looked at shared ancestral characters, so shared traits that originated in our ancestors. So if we're looking here for these characters, so we have a lobster, our outgroup, a silvertail, a dragonfly, a beetle, and a butterfly. We want to look at our outgroup, our most basic organism, and we can see that legs is found in all of these organisms we're interested in. Wing, development type, and mandibles, which are chewing, crushing mouth parts, are not shared with our outgroup. So those things, wings, development type, mandibles, are examples of shared derived characters. So they evolved later. And these are what are called synapomorphies. So we can make a character matrix with all of our characters we're interested in and go through and say which organisms have these characters. So we can say for legs, shrimp and lobster have more than 3 legs, but the rest of our group, so these are all insects, have 3 legs. So that's something that's shared between these 3, but not our outgroup. If we look at wings, wings didn't develop until for only these 3, so A does not have wings. The way that they develop is different for C and D compared to the other ones, and then mandibles, these chomping, piercing, crushing mouth parts are different for our butterflies. And so we have this character matrix, we can see changes in characters that occur over time. We can then use this to build a phylogenetic tree. So we take our character states and we figure out how organisms are related to each other. And we use the principle of maximum parsimony, which is choosing the simplest explanation or the most parsimonious of relationships for building a tree. So we want the fewest changes in our character state. So if we look here, we saw that 3 legs was shared by all of our organisms except for our outgroup. So that was easy. We already knew that one was our outgroup anyways. So it branched off first. So that is the character state that was different, that differentiated it. Then if we're looking at A through D, the presence of wings was the next thing that developed because B, C, and D all had wings. A did not, so that branched off. Then the way that they developed is different for C and D than it is for B, so B branches off. And then finally, C and D are different from each other because their mouth parts are different. So in this example, we had 1, 2, 3, 4 changes. If we look and try to add the changes in differently, this one has 5. 1, 2, 3, 4, 5. You could try doing it this way, and this one has 6. So we're going to hypothesize that this first phylogenetic tree is correct because the least number of changes had to occur for these organisms to be the way that they are today. So it's kind of like solving a puzzle. Any questions? For the OG? Because you could put it there, so this is saying that these organisms, A through D, all have 3 pairs of legs. So this one, that character state, you can always add a complementary character state. So if this is 3 legs, you could say this is more than 3 legs. Here's the development of wings. You could say here, there's no wings. So you can always add the complementary thing onto the tree, but we just usually add when something arises or if it disappears in one group of organisms. Okay.